So here we are in 15.2, and we're going to take a look at how pathogens are causing disease. And the first thing we're going to do, however, is take a look at Koch's postulates. Now these were derived in 1884 by Robert Koch. And what he did was he devised this approach for confirming some sort of relationship between disease and pathogens, or specific pathogens. So, back prior to 1884, people weren't really sure how they were getting the pathogen. They weren't really sure if it was a particular pathogen or what signs and symptoms were related to what particular pathogen. And so what Robert Koch did was he devised this approach, and so this approach needed to be taken in order to start to figure out what the signs and symptoms were for particular pathogens, and then that's what we now use. The information from this is what we now use in order to determine if somebody has a particular illness. So when we take a look at what Koch did was our approach is down below here. So he said what we're going to do is we're going to take a healthy organism, so we have our healthy mouse, we take a look at the red blood cells, and they're normal and fine, we don't see anything. And then when we culture some of the sample, some of the red blood cells, we don't see anything grow. Now, if we have a diseased organism, however, then we take that sample, so a blood sample, we can take a look at it, and we have something that's a suspected agent. So the suspected causative agent must be absent from healthy organisms, but present in diseased organisms. So his postulates, just like in geometry, says, okay, step one, if we have something that we're looking at, some sort of causative agent, what we have to do is we have to see that it's in each of these people that are diseased, but it is not in somebody that's not diseased. So then the second part is that our causative agent must be isolated from the diseased organism and then be able to grow in a pure culture. So what he said was, okay, all of the organisms that are diseased are going to have this agent in it, this causative agent. Those that don't have the disease won't. Now, the causative agent must be able to be isolated from the organism, and then you have to be able to take that, so a blood sample, for example, and be able to apply it to a pure culture, a plate, an agar plate, and then it has to be able to grow. And it has to not grow in the one that doesn't have the disease, the one that looks healthy, and we don't see this causative agent. Then number three is that cultured agent, the one that we have here now, that we cultured from the first infected host or organism, must cause the same disease in a healthy, susceptible organism. So then what they have to do is they say, okay, we took the diseased organism, we took a sample from it, blood sample, let's say, we see that it is in there, we can culture it on a pure culture or in a pure culture on an agar plate, and then we can take that organism from the agar plate and we can inject it into an otherwise healthy organism, and then we can see that they become diseased. So here's our healthy organism, and we can see that they become diseased. Then when they're diseased, step four is that same causative agent must be re-isolated from the inoculated diseased organism. So we then have to be able to take it out and do these steps here on the second organism. And then that is going to allow us to confirm a relationship between the disease that we're seeing with its signs and symptoms and a particular pathogen. Now, there are some limitations to this. So the first limitation is that this assumes that all pathogens can only be found in diseased individuals rather than in healthy ones. So we said that we have to be able to find this particular causative agent in somebody that's diseased and not find it in somebody that is not diseased. Now the problem is that we do find lots of different microorganisms in other people that are not actively infected or have any signs and symptoms or diseased. So for example, H. pylori is just found in the normal microbiota of the stomach. And sometimes when it overpowers everything else there and it multiplies and multiplies, then that's when it's going to cause chronic gastritis. So in that case, we couldn't say, okay, this person has a stomach problem, they have this causative agent, I think that's what's causing the stomach problem. Let's look at somebody that doesn't have a stomach problem, and then they swab the stomach, they get bacteria, and then they're able to grow that same organism. Then they wouldn't be able to say, okay, that organism is, or that person is diseased, and then that's due to this organism, because this other person has that same organism. So then that's a limitation. We just assume this. The second limitation is that it assumes that all healthy test subjects are equally susceptible to the disease. So if, let's say, we're that second mouse and we're otherwise healthy, and then we get an injection of this particular pathogen, it's just assuming that everybody's equally susceptible. Now, if my immune system is really functioning at a high level and you give me some sort of small regular dose of an infectious agent, then maybe my body can fight that off really, really easily. And so then I wouldn't get diseased, even though I did get that bacteria or whatever, and maybe it did actually cause the disease in the first person. If I'm healthy enough to fight it off and I don't get the signs or I don't get the symptoms, then that's a limitation we see by this process, because we're not going to be able to connect that causative agent to the disease of the first organism because the second organism just didn't get sick. This also is related to an individual's microbiota. So if you have a good, healthy microbiota in your intestines and your digestive tract and throughout your body, you've got really a whole lot of good, healthy bacteria and yeasts and things living inside of you. They create enough competition to where if you get some sort of pathogen in you, say on purpose, in these tests or just in regular everyday life, they're not going to be able to take hold. So by saying otherwise healthy, or assumes all healthy test subjects are equally susceptible, if I'm extremely healthy, then it's not going to be as susceptible. I won't be as susceptible to that illness as somebody that's not quite as healthy. Even if they're otherwise healthy, like meaning that they're not diseased in any way. But this can also be related to the competition inside of the person. And this is where we get the individual's microbiota varying. Also, the health of the immune system is going to vary. So two individuals may have different signs, symptoms, and severity of the disease. So when we're talking about its limitation, where we're saying all healthy test subjects are equally susceptible, we didn't take into consideration there that two individuals may have different signs and symptoms depending on the severity. So if we inject the first or the second person with the first person's sample, we may see some different signs and symptoms at different stages, and then we might incorrectly assume it was due to something else. The third limitation that we see is that it assumes that all pathogens are microorganisms that can be grown in a pure culture. And so remember we said that we take that organism out of the first human, for example, and then we are going to be able to see it, and then we can culture it. Now, this, again, is assuming that everything can be cultured, grown in an agar plate, but they can't. So, for example, something like an obligate intracellular pathogen, like viruses, some bacteria, we wouldn't be able to culture this, so we wouldn't be able to connect those dots. The next limitation, limitation 4, is that it assumes that all animals can serve as reliable models for human disease. So notice that we used a mouse in our example, or multiple mice in the example, and we do this and test these things out and see what happens, but it's still a guess because we are not mice, and so we then have to kind of infer some things, and we might be wrong. Another example, or an example related to this, is HIV only causes disease in humans. So we couldn't go through this process. We couldn't take a mouse or a rat and we couldn't inject it with HIV and see what happens because we are the ones that are susceptible and mice are not. And we can't do anything then about these Koch postulates because we cannot ethically inoculate or infect somebody with HIV. So we wouldn't be able to do steps 3 and 4 here. So then that's not going to be a good way to try to utilize Koch's postulates in order to identify and connect those dots. Again, another limitation. So since we do have these limitations, and also because our science has gotten more and more specific and more, I guess specific is a good word, we now have what are called molecular Koch's postulates. So this is a revised form of Koch's postulates by Stanley Falkow in 1988. And this is related more specifically to a gene that may cause an organism to be pathogenic. So rather than looking at the entire organism and determining if it's pathogenic and seeing the signs and the symptoms and connecting a specific organism to a specific disease, in this case what we're connecting to a certain type of pathogenicity or signs or symptoms are a specific gene. So there are still some limitations to the molecular Koch's postulates in that we can't genetically manipulate some pathogens, so it's not possible. And so then there are also some diseases that don't have suitable animal models. So let's take a look at molecular Koch's postulates here at the bottom. So on the left-hand side, we have just the general postulates. And on the right-hand side, they're talking about how that can be applied to EHEC, which is Enterohemorrhagic E.coli. So on the left-hand side here, number 1 is saying that the phenotype, so the sign or the symptom of a disease, should be associated only with pathogenic strains of a species. Now this is because sometimes people have a type of species, an organism in their body, but it's not pathogenic. And then we can see types that are pathogenic, and that's why the example over here is the EHEC, and that's because EHEC causes intestinal inflammation and diarrhea, but then we have lots of non-pathogenic strains of E. coli that are helpful and happy, and we want them living on our body because they're part of our microbiome. So the phenotype, the one that causes disease, should be associated only with the pathogenic strains. So again, this is separating them out, whereas before we would say like, okay, E. coli in Koch's postulate, we inject the E. coli and we look at the process, but now we're looking at something that's pathogenic specifically. 2 is inactivation of that suspected gene or genes as associated with pathogenicity, or that are associated with pathogenicity, should result in a measurable loss of pathogenicity. So if we can go in and we can knock out those genes, if we knock out those genes or that gene, then we should notice a difference in pathogenicity. So we should see a decrease in those signs and symptoms, the disease causing, or what is associated with disease. So on the example here on the right-hand side, it says one of the genes in EHEC encodes for the Shiga toxin, which is a bacterial toxin or poison, and that inhibits protein synthesis. So inactivating this particular gene can then reduce the bacteria's ability to cause disease. So we're seeing a decrease in the signs and symptoms, what's causing disease. And then 3 says reversion of the inactive gene should restore the disease phenotype. So we've knocked out the gene, but if we go back and revert it to its original form, then we should see that increase in pathogenicity again. So in the example, it says by adding the gene that encodes the toxin back into the genome, so with a phage or a plasmid, the EHEC's ability to cause disease is then restored. So again, this is kind of doing something similar to Koch's postulates, but it's more detailed. It's at the molecular level, where we say, okay, this is the phenotype. We can see the disease-causing genes or the pathogenic or virulence genes. If we inactivate those genes, we see a decrease in pathogenicity. If we reactivate them, we see pathogenicity again. So we've been using pathogenicity here. What is pathogenicity? This is the ability of a microbial agent to cause disease. So again, if it's a pathogen, we've said, that is an organism that causes disease. And therefore we have pathogenicity, is the ability for that microbe to cause disease. And then we also have virulence. So virulence is the degree to which an organism is pathogenic. So if we have an organism, we can say that's a pathogenic organism, and then we have different levels of virulence. So that's the degree that makes it pathogenic. And this is along a continuum. So we have things that are avirulent, so not harmful or asymptomatic, they don't have these symptoms, or low virulence, so to low virulence, which gives us just mild signs, mild symptoms, kind of feeling a little bit yucky, to highly virulent, so almost always causes disease, including multi-organ failure or death. So we have a continuum, and of course there's between all of these things as well. So everything from doesn't do harm and is asymptomatic, all the way up to likely will cause death or organ failure, and everything in between. So an example that's given in the text is inhalation anthrax. So in this case, the spores are inhaled, then they germinate, Then the infection causes toxins to be released. So now once they germinate, that means they're vegetative cells, which means that they are metabolically active. Then the infection causes toxins to be released. This causes fluid buildup. It decreases oxygen levels because we have an increase in fluid, we have a decrease in oxygen levels. This ends up causing death of different types of tissue, and then this can lead to organ failure and death of an individual. So inhalation anthrax is very, very dangerous, and we can compare that to something like the flu or to something like the cold or, something that's much, much lower, or say chicken pox, which causes a rash. Some of those can, of course, become dangerous, especially if we have other issues happening, immunocompromised or something like that. But typically they're mild or have low virulence. So when we're talking about virulence, then we need to have some way to quantify this, right? We can say that there's low virulence and high virulence, but what do we really mean? And where kind of are the cutoffs here? And the way that we can do that is by determining that with lab animals. And we have two different terms here that you should be familiar with that are related to virulence. The first one is our median infectious dose, or ID50, so infectious dose 50, median. So this is the number of pathogenic cells, or virions, that are required to cause an active infection in 50% of the inoculated animals. So that's where our 50 comes from. And when we're saying infectious dose, that means that it causes an infection, right? So we come up with a number, and in this case, what our number is the number of pathogenic cells, or virions, if we're talking about a virus. And this is the number required to cause an active infection, that's why it's infectious dose, in 50% of the inoculated animals. So if in a laboratory, it might be mice, might be monkeys or something, and then we can also apply this number to humans. The second one is median lethal dose. So LD50, lethal dose, and then median 50. So the number of pathogenic cells or virions or amount of a toxin that's required to kill 50% of the infected animals. So as the name implies, it's a lethal dose, meaning death. So the amount, again, we're going to have a number of pathogenic cells or virions that are required to kill 50% of the infected animals. So if we look at this on a chart here, what we see is something like this. We have our percent mortality in experimental group, and then we have the number of pathogenic agents, so the number of cells or the virions. And then we can take a look at this in a laboratory. So animals that are inoculated with, or animals are inoculated with known numbers of pathogenic cells or virions, so some particular type of concentration, and then that's plotted against the number of animals that are infected to give the infectious dose, or killed to give us our lethal dose, our median lethal dose and median infection dose. So in this case, what we're saying is if we give 0 pathogenic agents, then we have 0 that are either infected or die from the dose. And you can see that as we increase the number here, then we can increase the number of infected patients or killed patients. So then we go, and then when we get up to our 50%, so 50% of the population, if 10 are infected, that's of course a very low number, but if 10 were given some particular dose, so let's pretend it's this 10 to the 4, if they were given this 10 to the 4 cells of a pathogenic agent, and then 5 of them die, then that's going to be our lethal dose, our median lethal dose here. Or if we're talking about infectious dose, our median infectious dose. So when we talk about these infectious doses or lethal doses, they vary wildly. And the reason for this is many. There are many reasons. First of all, our route of entry, right? So in the laboratory, oftentimes they'll be injecting them. So they're getting right into the bloodstream, for example. But if we're talking realistically out in the world, we're not injecting, influenza into our bloodstream. So we're going to have a very different infectious dose. Also, age. So the age of the organism, if we're talking about humans, obviously we know that perhaps somebody in their 20s that's a relatively healthy person compared to even a relatively healthy person in their 80s is going to have a different dose that would cause infection, just because when you're 80, your body is just not working as well as when your body is 20. Also health, so I was just comparing age with two relatively healthy individuals, but health in general varies between somebody in their 20s or somebody in their 80s, within those age groups, I mean. So obviously we know that not everybody has the same level of health, and that would then, of course, affect the infectious dose. The immune status, so immunocompromised versus otherwise, environmental and pathogen-specific factors. So if we're talking about a particular pathogen, and say in a certain person, if we're talking about, let's say, H. pylori that we had earlier in the chapter, and we're saying that that can live in the stomach normally, but then when we get to these high numbers, it can cause this chronic gastritis. Say a person has a low amount of hydrochloric acid in their body, then it would be easier for H. pylori to infect the stomach and then cause that kind of chronic gastritis. And that just maybe is something that that person has always had. It's just a lower amount of acid, so a higher pH in their stomach compared to somebody else or maybe the general population. So those would be different factors to take into consideration as well, along with different environmental factors and pathogen-specific factors. So another thing to keep in mind, however, is that the infective dose does not necessarily correlate with the disease severity. So the amount of a pathogen that gets into the body and can cause infection in 50% of the organisms does not necessarily correlate with how severe a particular disease is. So for example, our S.enterica serotype typhimurium causes salmonella, so salmonella food poisoning. One single cell can cause an infection. So that is our infectious dose. Our median infectious dose is one single cell. So this causes nausea, can cause vomiting. However, it does not indicate its severity, right? Because salmonella food poisoning hardly ever causes death. So the mortality rate is less than 1%. So even though one single cell can cause the disease very easily, in 50% of the population, the mortality rate is very, very low. So it's not very severe. On the other hand, S.enterica serotype typhi causes typhoid fever. And in this case, it takes at least 1,000 cells to cause infection. So our median lethal dose here is 1,000 cells, which is quite a few cells. But in this case, it's severe and systemic infection, and the mortality rate is increased there up to 10%. So it takes a whole lot more to cause that disease, but that disease is much more severe in this case. So this is a table from the text. It is our ID50, so our median infectious dose for different foodborne illness or foodborne diseases. You don't need to memorize any of this information in here. It's just important to note here that if we take a look at the pathogen and the ID50 for them, first we have some viruses up here. And it's interesting, I think, to note that it can take between 10 to 100 for the hepatitis A virus in order to cause infection 50% of the people or organisms. But something like norovirus, which is kind of a typical virus that goes around, is only 1 to 10 viruses. But if we look down here, even with bacteria, same thing with rotavirus. That's kind of a common one, 10 to 100 in that one. However, if we look down here at bacteria and some protozoa, for Giardia lamblia here, it takes one single Giardia in order to cause infection. But if we look up here, look at our EPEC, or ETEC, the enterotoxigenic or the enteropathogenic E. coli, it takes so many bacteria for that to cause infection. So a person would have to have a heavy load of organisms in order to catch those, in order to get disease or be infectious. And then compare that to our enterohemorrhagic, which is 10 to 100 here. So I think it's interesting just to kind of notice the differences here. When we're talking about infectious doses, when we're talking about viruses versus bacteria versus protozoa, some of these are even around 10 to 100, while the viruses are as well. So really it's kind of all over the place, and it just really depends on that particular organism on when it's going to have the ID50, or when we're talking about an ID50 here, cause infection of 50%. So now we're going to talk about our primary versus opportunistic pathogens. So when we talk about our primary pathogen, this is, of course, one that causes disease in the host, but this is regardless of the host's resident microbiota or their immune system. So if we're saying that something is a primary pathogen, that means that it's going to cause disease even if the person's immune system is a healthy immune system, and even if that person has a healthy microbiome. So for example, the EHEC, the Enterohemorrhagic E.coli, produces this Shiga toxin, and this can lead to severe bloody diarrhea, inflammation, it can cause renal failure, and all of this can happen even in a person that's typically healthy. So if we look back at the number, remember it didn't take very many to get EHEC, and that would be in somebody that's otherwise healthy, so has a good healthy immune system, a good healthy microbiome that's protecting the organism, and it still will cause disease. So that is a primary pathogen where if you get it, you're likely going to get the disease, very, very likely. Something like Ebola, which is very infectious. If you come across it, you are likely going to get it. Now, when we talk about an opportunistic pathogen, on the other hand, an opportunistic pathogen is one that can only cause disease in situations where the host's defenses have been compromised. So in this case, we would have to have some sort of decrease in health level. We would have to have some sort of decrease in the immune system, for example, or maybe an unhealthy microbiome. So some of the defenses that we're talking about where situations have compromised them are, in general, the body's protective barriers. So perhaps there's a cut on the body. So if you were exposed to some particular bacterium and even just smeared it all over your arm, you would be totally fine and probably would not get infected. However, if on that arm you had a cut, now that body's protective barrier has been compromised, and then perhaps you would get the infection. And that's an opportunistic pathogen. Also with the immune system, so if you have a decrease in the immune system, it's an opportunistic pathogen. It's going to take that opportunity to cause infection and a decrease in the normal microbiome. So individuals susceptible to opportunistic pathogens, for example, are those that are very young, so they haven't built up their immune system very much yet. Those that are elderly, because their immune system just isn't working as effectively as it used to. Women who are pregnant, that's because their body's already putting so much energy into creating a new being. And since they are, then they're a little bit thinner as far as the immune system function. They've got a lot going on in there already, so things are a little bit compromised. Those that are undergoing chemotherapy, of course, if they're undergoing chemotherapy, chemotherapy kills our healthy cells as well as trying to kill the unhealthy cells or the cancerous cells. So then that would make a huge immunocompromised situation undergoing chemotherapy. Those with immunodeficiency, so in general, those that might have genetic diseases that relate to immunodeficiency, for example. Those that are recovering from surgery. So again, the immune system is being overwhelmed at the moment. So it's trying to go through and it's trying to heal itself from this surgery. And since it's healing itself from the surgery, then it's worn thin. So an opportunistic pathogen, that's their opportunity to come in and cause infection. Or with a breach of protective barrier. So like I said, a large wound, for example, or a burn, that's a severe situation where if a person has a very large burn across a percentage of their body, then opportunistic pathogen, that's a prime example of when someone might get infected with something that normally wouldn't cause infection. Like, for example, epidermidis, S. epidermidis. It typically is living on our skin and it's not a problem, but if we get a burn, for example, oftentimes that will get into the bloodstream and at high enough numbers it can actually cause infection, whereas typically it would just be all over the skin, and we want it to be on the skin because it's normal microbiome on the surface of our skin. Oh, that's my example right here. So S. epidermidis is part of the normal microbiota of the skin, but it can cause serious infections like endocarditis here. If it creates a biofilm, if it's on hospital supplies, or like I was mentioning, if there's a large portion of the body that's burned and then that S. epidermidis migrates from one area of the body to the burn site, then that can cause a serious infection and have something like endocarditis. So now let's talk about the stages of pathogenicity. So when we're talking about something being pathogenic, we have stages, just like we've broken other things down into stages. So these are the steps that a pathogen must achieve to cause disease, so to get us to that disease point. The first one, and we're going to talk about these in more detail, is exposure. So obviously you have to be exposed to cause disease. So contact, this is when they're going to gain entry into the host. Second, we have adhesion. This is when we have colonization. So it's going to stick to, adhesive, right? So adhesion, it's going to travel to the location and hang out where it's going to be able to establish infection. Then we have invasion, which is when it's going to evade or overcome the host's immune system, the immune response. And then we have infection. So this is when we're actually causing damage to the host. This is when we get our signs and our symptoms. So this cycle is complete then when the pathogen then exits the host and is transmitted to a new host. So our stages of pathogenicity are exposure, adhesion, invasion, infection, and then we'll see that transfer to a new host, and then all of those stages or the cycle is complete at that point when it leaves the host and moves on to the next one. So let's take a closer look at exposure or contact. So of course, this is when we encounter this potential pathogen. And we can do this in various ways. So via food that we eat, typical foodborne illnesses in that way, or things that we touch. So we kind of know that if somebody's coughing into their hands and then we shake their hands, that there's a high chance or a likelihood that you might get that illness, whatever they have. However, not all cause infection or disease. So it has to gain access into the host tissue. So even if somebody does cough or sneeze on their hand, and then they go to the door handle and open up the door, and then you later touch that door handle unknowingly, that doesn't necessarily mean that we're going to get the infection or that we're going to get that disease, because it has to gain access into the host tissue. So perhaps the microbiome on your hand is very healthy, and so it immediately is able to kill any of those pathogens. And then you're fine and you're none the wiser. So we do have different portals of entry. And these portals of entry are specific anatomical sites through which a pathogen can pass into the host tissue. So one major portal of entry are mucosal surfaces. So this is our most important portal of entry for microbes, because if we think about our mucosal surfaces, this is kind of how we think about ourselves getting sick anyway, is through our respiratory tract, so breathing in pathogens. So if somebody's coughing, for example, you kind of think like, oh, I don't want to breathe around them, because you might breathe in those pathogens that they are coughing out of their respiratory system. The GI tract, so this relates more specifically to the food we eat, for example, in foodborne illnesses. So if we have some sort of bacteria growth on the food that we have in front of us, and then we eat that food, our mucosal membranes, specifically membranes in the GI tract, are an important portal for that. They can burrow their way through those cells in the intestinal tract and cause infection elsewhere, or in the intestinal tract. Our genitourinary tract, so when we're talking about our genital tracts and our urinary tracts, so those are open to the exterior. So when we're talking about our mucous membranes, oftentimes we're talking about things that are open to the exterior but are kind of interior to the body. So things like our eyes, if we are touching something that has some sort of pathogen in it and we rub our eyes, for example, we can transfer that pathogen into our eyes, and then it's going to be in the body. It can get through that mucous membrane. Our nose, our mouth, we kind of already know those things with breathing things in, or through our nose and through our mouth. Through the urethra, so coming in through the urethra, that's how urinary tract infections, for example, can start, is coming up through the urethra, and then also through the anus, so anything that's open to the exterior, so then we can cause some intestinal infections there. We do have what's called portal specificity. Not we, but pathogens have portal specificity. So these are where we have different pathogens are suited for different portals. So this is based on the organism's environmental adaptations. So how that organism infects a particular organism, so infects the host, is what's going to allow it to be to have that specific portal. So portal specificity is if a particular pathogen is more well suited to infect the host in a particular way. So like I was saying before, if we have it on the surface of the skin, maybe some organisms do really well with infecting through the surface of the skin because they have particular enzymes that break down some of the barriers that we have on the surface of the skin, whereas others might not. However, the others might get into the body in a particular way, through a mucous membrane, and then that's an easier way for them to get through. And this is based on their environmental adaptations. So if they have been in an environment, maybe they can learn how to easier get through that environment. Also, it depends on the enzymes that they make. So like I mentioned earlier with Helicobacter pylori, H. pylori, It's going to make that urease that's going to protect itself from the pH of the stomach, the low pH, the acidic stomach, and then it's going to be able to burrow its way through the cells that are in the stomach and the toxins that they secrete. So if they secrete a particular toxin that needs to go into the bloodstream, if they're not in the bloodstream, then that's not a good portal of entry for them. So they have a specific portal that would be better for them, like getting into the bloodstream. Then we have our parenteral route, and these are pathogens that enter via breach in the protective barrier of the skin and mucous membranes. So this is how we oftentimes think that people are getting sick, is through these ways. And the reason we think that is because that is oftentimes how people get sick. It's through these mucous membranes, so breathing things in, eating things in, and then also they're able to breach this protective barrier of the skin, and likely that's because of a burn or a cut or something. Now, when we're talking about exposure to a fetus, for example, we have a different situation here because typically, of course, we're not talking about when a fetus is growing that they're going to have exposure, that they touch things or they breathe in things typically. So when we talk about exposure for a fetus, because they can get infections as well, we have pathogens that are capable of crossing this placental barrier. So in a pregnant woman, the placenta is normally going to prevent those microbes from passing to the fetus. So if a pregnant woman gets sick with something or infected with something, oftentimes the placenta is going to prevent that from getting to the fetus. But some things are able to cross that blood-placental barrier. So some examples are Listeria monocytogenes, and torch. So torch is going to stand for these items here, torch infections. So our T is our toxoplasmosis, which maybe you already are familiar with, saying that when a woman is pregnant, that they shouldn't be cleaning kitty litter, for example, because toxoplasmosis oftentimes comes from kitty litter or cat feces. The O here is going to stand for this group here. So syphilis, chickenpox, hepatitis B, HIV, and then Fifth's disease, which is something a little bit different. All of these are going to come from these pathogens here. So that stands for, is the O portion. The R is rubella, so German measles from the toga virus. C is cytomegalovirus, which is human herpes virus 5 is the pathogen for that. And H is for herpes, so the herpes simplex virus, both 1 and 2. So all of these, the TORCH, you should be familiar with those. The torch, those are the ones that can go through the blood placental barrier and can infect the growing fetus. We have transmission that can come from the mother to the baby also during birth through the birth canal. So these here, the torch and the listeriosis, are those that can go through the blood placental barrier. And then we also have to keep in mind that at this point it's not a fetus, but it's a baby, and the baby can get an infection as it's going through the birth canal. So things like chlamydia or gonorrhea can be passed along to the newborn, and those things can actually cause newborn blindness, which is why a long time ago people started putting antibiotics in the eyes of a newborn. Nowadays, you don't have to do that, and you can opt out of that type of thing because you're already giving antibiotics and killing bacteria in a newly born baby, which might not be the best idea because at that time they're trying to build up their immune system, and if we're already giving antibiotics at that point, then that can cause some damage, starting from a very young age at birth. And moving along, it might take longer for them to build up their microbiome. However, if a mother does have chlamydia or gonorrhea, that would be an important first thing to do because you, of course, don't want to cause blindness in your child. So there are some tests that are often done on pregnant women ahead of time to see if they have some of these diseases that can be passed while the baby's going through the birth canal, and then they can take measures to prevent that from causing disease in their child. So now we're moving on to our second stage. We've had exposure, and we've talked about the different exposures and different portals of entry. And now we're going to talk about adhesion. So this is the capability of the pathogen to attach to the cells of the body using what are called adhesion factors or other mechanisms. So that's why we have adhesion. Some of these adhesion factors, these are called adhesins. These are typically proteins, but also carbohydrates that are on the surface of certain pathogens. And these are used to bind to specific receptors. So oftentimes they are glycoproteins, and then they're going to be attaching to specific receptors on our host cells. So an example of this, an adhesin, would be like when we've been speaking about HIV, for example. HIV has a glycoprotein that's going to attach to that CCR5 receptor, and then it's going to cause a change, or the CD4 receptor, and then cause a change and attach to the CCR5 receptor. And that's because of the glycoproteins on the surface of that HIV virus. And that would be considered an adhesin. And we see these also on the surface of different bacteria, on different other microorganisms that we can talk about, different protozoas and things like that. So, for example, we also have other mechanisms that aren't specifically glycoproteins on the surface. So also think of glycoproteins like these glycocalyces. We have slime layers and capsules, these kind of sticky things that allow them to attach. In protozoas, oftentimes we see things like these hooks and barbs. So remember back to our Taenia, our different worms, our helminths, for example, also that have hooks, if you can think of those hooks, and we see those on different protozoa. We see spike proteins on viruses, so that's what I was saying with the glycoproteins on the HIV virus, those spike proteins. So we have hooks and barbs on protozoa, we have spike proteins on viruses, we have glycocalyces, so including slime layers and capsules that are on bacteria that make them very sticky and allow them to attach. And then that is going to allow them to get into the body further. But first we have this stage of adhesion, so the sticking of the pathogen to some particular portion of the body. This is where we can also remind ourselves about biofilms. So remember the biofilm, we have a glycocalyx, this EPS, this extra polymeric substance that's being secreted by these bacteria, and it's creating this matrix that is going to allow those bacteria to attach to certain surfaces and then grow to a large number within these biofilms. And we saw our video here previously that we have on the P. aeruginosa biofilm. So the biofilm of P. aeruginosa is common in those with cystic fibrosis, burn wounds or middle ear infections, and it makes it much harder to remove that pathogen because of this biofilm production. We've talked about that previously, why biofilms are the way that they are and why they're so difficult to get rid of. But in general, just a reminder that they're harder to remove the pathogen because it's harder to get them to get out of there mechanically, washing them away or getting rid of them, and also via chemical means. Because remember, antibiotics can't reach the bacteria that are inside, or in the biofilm, they're not actively growing, and a lot of our antibiotics attack the actively growing bacteria. So then we have invasion. So we have been exposed to the pathogen has now attached on, so we have these adhesins, for example, or other mechanisms that attach or allow it to stick to us, so adhesion. And now we're moving into invasion. And invasion involves this dissemination of a pathogen throughout the local tissues of the body. So it's invaded now. It's attached, and now it's going to move further throughout the body. And they can do this utilizing things like exoenzymes or toxins. So the example that I've used previously also with H.pylori, when it is in the stomach here, we have our epithelial cells of the stomach, and then within that we have our mucin gel, which is produced by specific cells of the intestine or of the stomach. So they secrete this to protect the epithelial cells from the hydrochloric acid, the low pH that's in the stomach. But our H. pylori is able to make urease. When it makes urease, it allows it to burrow through that mucin gel. So not only does it increase the pH so that the low pH doesn't kill the bacteria, but it also can now burrow through this gel and then get into the cells so that it can get further into the body. So it neutralizes the stomach acid, and this causes that mucin to liquefy, and then it can swim right through it and get into the cells. So this would be invasion. So this is an exoenzyme that it's making. Its virulence factors are going to allow those pathogens to colonize and then damage the host tissues as they get deeper and deeper in the body. And then, as in this example, some virulence factors are going to protect the pathogen from the immune system defenses as well. So we have the exoenzymes or the toxins that are going to allow it to degrade different things, like this example, the mucin gel, but also to degrade things like we have the phospholipases or something. that are going to be breaking down phospholipase, and that is, of course, a major part of our plasma membranes. So if an bacteria, for example, is making phospholipase as an exoenzyme, it can start breaking down those phospholipids on the surface of the cell, on the plasma membrane, and it can burrow its way through our cells to get into the body and then invade the body. Also, then we have intracellular pathogens. So we just spoke about things that make exoenzymes or toxins in order to get and invade the body. An intracellular pathogen, or for an intracellular pathogen, they have to achieve invasion by entering the host cells and then reproducing. So again, we have our obligate intracellular pathogen. So it's obligated to, it has to be inside of the cell, and it's a pathogen. So again, it only reproduces in the host cell. So this is our viruses, for example, our obligate intracellular pathogens. We also have facultative intracellular pathogens. So these can actually reproduce inside the cell or outside of the host cell. So facultative, similar to when we think of facultative, it can switch and it can change, and that's the case here. So it doesn't have to be inside of a cell, but it can be inside of the cell. So in order to do this, they can evade some mechanisms of the immune system, and then they can exploit the cell's nutrients. So in order to get into the body, in order to get into the cells, our intracellular pathogens have to somehow evade the immune system so that they don't get killed before they even get to the cells. And then they can also exploit that cell's nutrients. So they can utilize the nutrients of the cell. They have to evade the immune system. And we see this even when they get into our cells. So they have to evade the immune system as they're traveling through the body and getting into the cells. Then once they're in the cells, they want to evade the immune system because they need time to reproduce. And then they can take that time by exploiting the cell's nutrients, so utilizing the energy or the ATP that's been made by that organism already. And we've talked a lot about viruses already, so you already know how it kind of utilizes and takes control of the host machinery and utilizes the resources within it. So when we have invasion, right, remember we're getting into the cell, so we've already spoken about things like exoenzymes and toxins, and then we have our intracellular pathogens. Now they have to get inside of the cell in some way. So they can enter by endocytosis or by phagocytosis. So if we look at entry by endocytosis, this is when we have effector proteins are going to be secreted by the pathogen, and it's going to trigger the cell to bring it into itself. So for example, Salmonella secretes molecules that are going to cause protrusions in the intestinal cell membranes, and then that brings the pathogen into the cell. So these protrusions are kind of like wavy parts to the plasma membrane. And then when it does this, it actually is going to grab onto the bacteria, and then it brings it into the cell. Brings it into the actual intestinal cell, and then that's where we get our intestinal tract issues. Or here we have surface proteins that can bind to the receptors in the host cell and then gain entry. So an example here is Yersinia here produces protein invasion, so an invasion protein, and that binds to the beta-1 integrins on the host cells. So it's kind of a receptor, a lock and key mechanism. Similar to what we've spoken about with HIV, where it attaches to the CD4 receptor, the CCR5 receptor kind of initiates further, and then it brings it into the cell. So the immune system cell just normally brings HIV into the cell through endocytosis, and this is similar. But the other example here is the Yersinia here that makes this invasion protein, and then that binds to the integrins, and then the host cell brings it in via endocytosis. Or when we get entry through phagocytosis, this is where our white blood cells, as they normally do, they're going to phagocytize cells. So they go around looking for things that are not supposed to be there. They find one, they attach onto it, they grab it and bring it into the cell, into the white blood cell. And the intention is to then destroy the bacteria or the pathogen inside of the white blood cell. So the white blood cell is going to phagocytize to destroy the bacteria by binding to the phagosome with the lysosome. And then this is when we are going to release digestive enzymes that are supposed to degrade and break down the bacteria. Now, some examples that we have here through phagocytosis is that Shigella actually lyses that phagosome prior to it binding with the lysosome. So our white blood cell is going about its business. It comes upon Shigella, and it says this isn't supposed to be here. So it actually is going to grab onto that Shigella bacteria, and then it's going to bring it into its phagosome, because it's the white blood cell is a cell that goes through phagocytosis. When it goes through phagocytosis, it's grabbing onto it, bringing it into itself, and when it has that little pocket with the bacteria in it, that's called a phagosome. It brings in Shigella into that phagosome, but then before the phagosome binds to the lysosome, and remember the lysosome has these digestive enzymes, before it binds to the lysosome, it gets out. So it lyses that phagosome, and now it's within the white blood cell, and now it can go through its process of multiplying and creating its virulence factors. Another example is M. tuberculosis. It actually prevents fusion with the lysosome. So it also is brought into the white blood cell. It's in a phagosome, but then it prevents fusion with the lysosome, and then it multiplies within the phagosome itself. So it allows itself to get brought into that white blood cell, and now that it's in the phagosome, it's kind of protected. It actually prevents that lysosome from binding, and it can make many copies of itself. So through endocytosis is where it kind of tricks the cell to bring it into the cell. Either by utilizing these surface proteins to bind to receptors, or to somehow adjust the cell to make it want to bring something in, or through phagocytosis, when the white blood cell is actually doing its job of gobbling up things that aren't supposed to be there, but once it gobbles it up, it has it in the phagosome, and then our different pathogens can go about evading that immune system. So now we're moving into infection. So infection is our successful multiplication of the pathogen. Once it has successfully multiplied, remember when we only have a certain number, then that's not considered an infection. It's only once it's colonized, once there's successful multiplication of this pathogen, that then we would call it an infection. Now we have different types of infections, however. So the first type is our local infection. And as its name implies, that's when it's confined to a small area of the body. That's typically near the portal of entry. So wherever it got into the body, then we have a local infection there. So for example, a UTI is confined to the bladder. It's a local infection. Or if you get a cut and that cut, gets all red and puffy and things, then that's a local infection. It's not something getting into the bloodstream and spreading throughout the body. We also have a focal infection. Focal infection is a localized pathogen, or the toxin it produces is going to spread to a secondary location. So we have a local infection, but then that local infection is going to spread to a second location. So for example, if we nick our gums while we're brushing our teeth or while we are flossing, that can lead to streptococcus of the oral microbiota traveling through the bloodstream to a different location. So it normally is in the mouth, and when it's normally in the mouth, it's doing just fine in this case, so it's not even a local infection, as in a pathogenic bacteria, but then it can travel through the bloodstream to another location and then cause problems in that location because it's not supposed to be there. And then lastly, we have our systemic infection. So this is when an infection is becoming disseminated throughout the entire body, so throughout the system. And some examples there would be the varicella zoster virus. So it gains entry in the upper respiratory system, and it spreads throughout the body, and then it causes chickenpox. So we end up seeing that throughout the entire body. Another type of infection is a secondary infection. So when we talk about a secondary infection, this, of course, means that there's a primary infection, and the primary infection is the initial site of the infection. So wherever that infection started is obviously the primary infection. And then that primary infection by one pathogen can lead to an infection by another pathogen. So a secondary infection is not that it's moving and going to another place, because we already talked about that as a focal infection. A secondary infection means that there's a primary infection with one pathogen, and then there's an additional infection by a secondary pathogen. So, for example, the immune system of a patient with HIV infection is immunocompromised, so they have their primary infection, which is by HIV, but then they're more susceptible to a secondary infection. So oftentimes we see secondary infections like oral thrush or some other pathogen in somebody that has HIV because their immune system is compromised, so they have a secondary infection as well. Some secondary infections, however, are the result of the treatment of the primary infection. So, for example, if somebody has a primary infection, they go into the doctor or the hospital or something, and they're given an antibiotic. That antibiotic is going to target that primary pathogen, but it also is going to kill the organism, the human's normal microbiota. When it does this, then there's no longer that competition in that microbiome. Having that healthy microbiome is no longer there to help protect that person from another pathogen. And so then this creates that opening for those opportunistic pathogens. Remember, if we have a decrease in the health of the microbiome, then we leave ourselves available to an opportunistic pathogen. And so then that would be a secondary infection. So the primary infection, the person is taking antibiotics for, it might kill that primary pathogen, but at the same time, you might be getting a secondary infection. So then our last one is our transmission of the disease. And so this is related to our portal of exit. So we had our portal of entry is how it got in, and we have our portals of exit. This is the location a pathogen leaves the host to be transmitted to the new host. And our pathogens are adapted to use specific portals of exit, just like they are adapted to use specific portals of entrance or entry. And we see a lot of the same places, like respiratory, for example, right? So again, people are coughing. As they're coughing, that would be a portal of exit. So we would, get viruses or bacteria spread that way or transmitted that way through the GI tract. So as you have an intestinal infection using the restroom, and then another person using the restroom, or using the restroom and not washing your hands, for example, and then transferring that to somebody else would be a portal of exit. And the urogenital tracts, again, so transmitting through the urinary system or the genital system, reproductive system, is then another location. All of these different areas that are portal of entry are also portal of exits. So again, through coughing, sneezing, different secretions, feces, urine, semen, vaginal secretions, tears, sweat, shed skin cells. If it's an infection that's on the skin, the shed skin cells can contain the pathogen and then pass that along to others. So that could be a portal of exit as well. And then remember that pathogens also rely on insect vectors for transmission. So they'll exit the body via the blood that's extracted by a biting insect, or blood extracted by needles and transmitted that way as well. So moving from a person's blood. So even if it's a type of pathogen that isn't typically infecting the respiratory system or the GI system, for example, if it's something that's in the bloodstream, it can still be transmitted. So via blood extracted through an insect or through needles. All right, and that wraps up our 15.2.